Hello, we are in our last set of notes for this unit as well as this course. We're going to go over fertilization and pregnancy. You do have a handout. I've given you a template that you can fill out, and that should make things a lot easier. I have also, this time around, added the diagrams directly into the handout as well. So, as usual, when those diagrams pop up, you're welcome to pause the slides and fill in any of those labels. If you find that the diagram is not very clear on your mobile device, you'll also find them in a textbook. And so you can use that as another resource if that will help instead. We have 4 topics that we're going to cover today. We're going to go over fertilization, embryonic development, so the development of an embryo, followed by development of a fetus or fetal development, and labor and childbirth would be the last thing that we're talking about as well. Okay, let's get started. So when it comes to fertilization, I want to just quickly bring up a couple things that we talked about in the last set of slides. Remember that when an oocyte is released from an ovary in ovulation, that oocyte is actually viable for only a short amount of time, up to 24 hours after ovulation, that oocyte is viable for fertilization. In addition to that, sperm also have a pretty small window that they're viable for, and that is 12 to 48 hours after ejaculation. So you're faced with those 2 small windows of time that these 2 gametes are viable if they're going to be able to come together and successfully produce a fertilized cell. In addition to that, however, sperm are also faced with the added challenge of having to travel quite a distance. So ejaculation happens down in the birth canal, near the inferior part of the uterus and the vagina and everything. And so those sperm cells have to travel all the way across the uterus, then into the fallopian tubes, and make it all the way to practically the end of those fallopian tubes, the distal ends of those fallopian tubes, where these oocytes are traveling at a much slower pace than they are. And so if you add that challenge on as well, and take that travel time, I guess you could call it, into account. There are people out there who are able to figure all this out in terms of timing and everything. When a couple are trying to get pregnant, these things are all taken into account, and so the time window for fertilization to occur is generally no more than 72 hours before and no later than 24 hours after ovulation. So that's kind of the window, the general window given to people if they're trying to get pregnant and have a baby, that they can, make attempts to try and have a successful fertilization. Okay, in order for fertilization to occur, there are certain things that need to happen to make sure that given that there's a very small window of time, these things are in place to kind of make the process happen as efficiently as possible. So when sperm first enter the birth canal, enter the uterus, they need to pretty much orient themselves immediately so that they start swimming in the direction of the primary objective, which is that oocyte. So sperm are actually chemically attracted to the oocyte. There are certain chemicals that are present in the outer membrane, outer layer of the oocyte that will attract sperm, so it's almost like a chemical homing device for them. And so they'll head immediately towards that superior part of the uterus and then eventually figure out where the fallopian tubes are and head in those directions. And so when they finally encounter an oocyte, the sperm have these structures called acrosomes, or an acrosome, at the end of kind of like a nose for the head of each of these sperm cells. And that acrosome contains special enzymes in it. And so that acrosome breaks, ruptures, releases these enzymes, and these enzymes will actually start to break down the outer protective layer of the oocyte. And so several sperm that kind of reach the destination at the same time, all of their acrosomes break, and the enzymes that are released break down that protective layer. And then one of those sperm Not necessarily the first one that arrived at the oocyte, but one of those sperm will just happen to be able to break all the way through that layer, that outer layer, and will come in contact with receptors that are in the oocyte's membranes, the outer membrane. And as soon as that contact happens, that sperm that breaks through is pulled into the cytoplasm of the oocyte, and simultaneously, all the other sperm cells are blocked basically. They're not going to be allowed to enter into the cytoplasm of the oocyte as well. Now, there sometimes are fluke situations that happen where more than one sperm cell might break that outer layer at the same time, and so those could eventually lead into possibly like twins and so on. But for the most part, once one breaks through that outer layer, all the others that are trying are not going to be successful in getting through. Okay, so the process of fertilization itself, by definition, is when the genetic material of a sperm combines with the genetic material of the oocyte. And before fertilization happened, that oocyte, after it was released from the ovary in ovulation, kind of goes into a frozen state. It's not able to continue all the way through its meiosis divisions. But as soon as that fertilization happens and it combines with the genetic material of the sperm, that oocyte will finish up those divisions, meiosis II divisions, and will turn into something called a zygote. And from this point onwards, zygote up until the 9th week of development in the uterus, this mass of cells, as it's changing and growing bigger, going through many more divisions and so on, it's going to be called an embryo. Okay, so the zygote, when you take the genetic material of a sperm, genetic material of an oocyte, and they combine, it's almost like two halves of DNA come together to make a whole. So a zygote is actually the first one cell of a new individual. And this zygote exists in the fallopian tube, right at the distal end, wherever the fertilization process happened. And it'll continue to travel through that fallopian tube towards the uterus. Now, as it's traveling at a very slow pace, it is going to go through a set of divisions. And these divisions are called cleavages, and they happen very quickly, one right after the other. As soon as one cleavage is done, another one immediately happens. And it's really just a matter of that one cell goes through a first cleavage and turns into two. These are mitotic divisions here. So the one cell turns into two. Immediately, those two each split up into halves again. And then you have four. And then those four become eight, and so on and so on. And it happens one immediately after the other. Now, these daughter cells are very small, and they're small because these divisions happen so fast that there is very little growth of those cells between each cleavage. And so as these divisions are happening, you end up with more cells, but these cells get continuously smaller and smaller in size compared to the previous divisions. Eventually, when you have about 16 cells, after a couple of these cleavages, about 16 cells, that ball of cells is called a morula, and that is the form that it's in right when it's reaching the end of the fallopian tube, or I should say the proximal end of the fallopian tube, and is entering the uterus. Then a few more divisions happen, and it turns into something called a blastocyst. And that blastocyst is the form that it's in when it implants into the uterine wall. Okay, here is your first diagram. So at this moment, right now, you can actually pause this video and go ahead and fill in those blanks. Okay, so let's go over these diagrams. This diagram real quickly here. Along the upper part of the diagram, you can see the different stages of that zygote as it goes through its divisions, its cleavages. And you can see that each of these stages has a label. There's A, B, C, D, E. And those letters correspond to labels that are on the lower diagram, which is the female reproductive tract there. You can see if we start at the ovary, there's ovulation that takes place. The secondary oocyte that's released is going to encounter some sperm cells. And at A, that's where a zygote is formed. So that's fertilization. That zygote then goes through one cleavage, two cleavage. By the time you're at B, you have an early cleavage 4-cell stage. And then it'll keep going, cleavage, cleavage, and then at C, you have a morula. And that is right around the time that that mass of cells, that embryo, is entering the uterus. Then you have D, that is an early blastocyst. And you can see blastocysts actually by comparison have this cavity or hollow in the middle of them. And that hollow is important. We'll talk about in a second here. But there you have an early blastocyst. And then at E, that blastocyst has turned into, the early blastocyst has turned to late one. And that form that you see there is the state it's in when it implants into the endometrium, that inner lining of the uterus. Okay, let's talk a little bit more about what goes into what's going on inside of that blastocyst. So the blastocyst is a ball-like circle of cells. Approximately 100 cells are in a blastocyst. And inside of that blastocyst, the majority of it inside is rather hollow, but there are also these what we call the primary germ layers that are eventually formed inside of the blastocyst. And these are layers of tissue that will kind of are the first beginnings of some major tissues and areas of the body. There are 3 primary germ layers, and you have them as bullets on your handout here. So the first is the ectoderm, and the ectoderm is the outside layer of the 3 layers. This is the layer that's eventually going to turn into the majority of the nervous tissues, brain matter, neurons, and so on. Also, the ectoderm turns into the epidermis, or outer layer, most superficial layer of the skin. Then you have the mesoderm. This is the middle layer. This will turn into mucosa, which are a lot of the inner lining membranes of the body, as well as a lot of the glands of the body, both endocrine and exocrine. Okay, and then the third primary germ layer is the endoderm, and this is the inside layer. This layer is going to become pretty much everything else that is not coming from ectoderm or mesoderm. Okay, so as these germ layers develop, this blastocyst will, continue to change in shape and go through more divisions internally and so on. And then by the time you have a late blastocyst, it implants into the wall of the uterus, and that's typically around day 14 of the ovarian cycle. Okay, after implantation happens, there are a few things that are developed soon after that are going to help that blastocyst eventually turn into a bigger embryo, then a fetus, and so on. So the first are what we call chorionic villi. These are actually just extensions of the blastocyst. So they're projections that will stick into the endometrium, the endometrial layer of the uterus. And these chorionic villi actually work with those cells in the uterine wall and form something called the placenta that we'll talk about in a second here on another slide. But they help to form the placenta. After implantation, there's also the development of something called the amnion. That is a fluid-filled sac that surrounds the embryo. It'll provide protection and kind of like a watery cushion from, to protect that embryo as it grows. It provides a medium that has lots of electrolytes and other dissolved materials that will help to allow that embryo to develop in terms of its tissues and so on. And then the third thing that is developed after implantation is the umbilical cord. That is going to attach the embryo to the placenta and eventually be a way for that embryo to gain certain materials and excrete certain materials um, to its mother and eventually out or in from her system. Okay, here you have your second diagram. Go ahead and pause at this time and fill in the labels, which are primarily to the right of the diagram. There aren't any on the left. Okay, so let's go over the diagram. On the left-hand side, it looks kind of like a weird pink light bulb. That's actually the uterus. You can see that the outer layer is a bit darker, but the inner layer is lighter pink. That is the endometrial layer. Okay, the bottom part of it, that's the cervix and the birth canal. But towards the top, you can see the orangey red structure is what we're zooming into towards the right of this diagram, and that is the implanted blastocyst and what is turning into an embryo. So let's kind of work our way from the inside to the out. So on the inside, you can see that there are 3 layers there. These have been stained so that you can see them a bit clearer. But there's an upper layer, that's the ectoderm. Then you have the mesoderm, which is pinkish. Ectoderm is kind of blue. And then yellowish underneath, that's the endoderm. So these are the 3 primary germ layers. Then surrounding those layers, which is essentially the embryo, that's going to turn into the fetus, the developing fetus. But the amnion is what looks like empty space is actually filled with fluid above and below that embryo. Okay, and then towards the right of the embryo, you can see there's the start of the umbilical cord, and that will eventually attach to the placenta. This is a bit too early to see the placenta being formed there, but we'll see it on another diagram later on in these notes. And then if you look around, kind of radiating from the embryo, you can see these extensions, and these are the chorionic villi. So these kind of projections, almost like rays on a sun, that kind of looks like that. These are the chorionic villi that are eventually going to work with the cells that are in the endometrial layer and help to anchor the embryo as it grows and continue to provide all the things that it needs, like in terms of building blocks, materials to allow that embryo to grow into a fetus and so on. Okay. All right, let's go over the placenta. So the placenta actually, although it it's technically part of both the mother and the embryo, it actually forms kind of a barrier between the two. Blood is not exchanged between the mother and the embryo. So it's not like we have one continuous cardiovascular system that goes into the embryo, out the embryo, into the mother, and so on. There is a distinct separation between the mother's cardiovascular system and the embryo's cardiovascular system. Okay. However, the placenta does provide 3 functions for the growing embryo and its mother. First, it's going to allow a way for the mother's cardiovascular system to let nutrients and oxygen make its way to the embryo. So it's almost like a little gateway. Secondly, if there are any wastes that are, well, not if, there are going to be wastes that are produced as the embryo grows and develops, and so these wastes have to be excreted, and so they will exit the embryo through the placenta, the umbilical cord and placenta. And then the third is that the placenta acts as an endocrine organ. It is going to be producing hormones that are going to help maintain that pregnancy. Certain changes have to happen physiologically to the mother to, as that baby's growing, to support that baby. So changes physically to her shape, to her body. Certain hormones need to be secreted to um, to make all these things kind of work properly. So especially estrogen and progesterone, these are going to be secreted by the placenta and help both mother and growing baby as they develop. Okay, we're almost done here. We should be on the last page of these, the handout. So after, let's say we go through the first 9 weeks of development, that is, during that time, it's called an embryo, but after that, we now consider it a fetus. Okay, so that embryo, by the time we reach the end of the 8th week, all of the major organ systems are, for the most part, fully formed in that embryo. And so by the beginning of the 9th week, we don't call it an embryo anymore, we now have a fetus. And from this point onward, the changes that are going to happen, now that the groundwork has been laid, the changes that are going to be happening in that fetus are more towards growth. Okay, so just, increasing in size. The organ systems that are already there now are going to be developed more in terms of being specialized for their actual jobs. And there's also going to be changes that happen in terms of body proportions. Okay, so this will happen for the remainder of the time that the fetus develops inside of his or her mother. And so approximately 270 days, which is roughly 9 months after fertilization, we now consider this fetus as full term. And we can move on and briefly talk about the stages of childbirth. Okay, so childbirth is also known as parturition, or sometimes so childbirth and parturition are the same thing, but the stages that are involved in the process of childbirth or parturition are known as the stages of labor. So these are a series of events that take place in order to expel the infant from the uterus, the fetus from the uterus. Okay, so there are 3 main stages that we're going to talk about, and I'm going to go through these really, really generally because you're going to be doing an activity that will look into these a bit more in detail on your own. So just generally speaking, there are 3 stages. The first is the dilation of the cervix. Second, expulsion or delivery of the infant. And eventually, third is delivery of the placenta. Okay, and this is the last slide, so you're welcome to fill in those missing labels. There aren't too many, but you will notice that, so there are no labels on the diagrams themselves, just places where you should label the 3 stages. So dilation of the cervix, expulsion, and then delivery of the placenta. Okay, and that's it in terms of notes. So when it comes to the stages of labor, once these notes are taped in, you can set up the next page and actually work on going through and using the textbook to go through the stages of labor. Okay, thanks you guys.